Okay, thank you, Dr. Chen. I'm going to be using a timer to measure my time, although our system, our bodies, many organisms, all organisms know how to measure time. And what I want to talk about today is how we're using fungi to understand the molecular mechanisms behind time measurement, but actually how an organism as simple as a fungus measure light in a very accurate manner to have that clock working very precisely in sync with the environment. So I always wanted to start a talk with this phrase, and I think this is the place to do it. So if you look at the Bible, if you look at many cultures, we want to know how life started, and we know that life in our planet has been exposed to a very rhythmic environment. Since the beginning of time, we have been exposed to days and nights. And that's beautiful, provides beautiful sunsets, beautiful sunrises, but it is very stressing for organisms. If you are outside, that means that UV radiation is going to go up, temperature is going to go up, humidity is going to go down, and you might desiccate, or the light itself might kill you, as our colleague was telling us here. But if you know when the sun is going to come up and you can be prepared for that, you're going to be in a much better position. And this is the context where circadian clocks emerged. Many times throughout evolution, as convergent evolution, clocks have emerged. Why? To allow organisms to know time and to anticipate daily changes. So basically within our cells, we have molecular devices that can measure time in a very precise manner. And very importantly, these clocks get synchronized to the environment through light signals. The light is telling us every day, yep, you were right, this was the time to get up, live your day, then go to bed. Next day, the light once again tells us, yep, correct, you should get up. And not only getting up and going to bed, clocks are controlling a plethora of things in organisms, gene expression, physiology. If you look at gene expression, close to 40% of the genome can be cycling in a given organism. Other numbers say that if you combine different tissues, maybe 90% of the genome can be oscillating in a daily fashion. So clocks are conserved from fungi to mammals. And you see that the names of the components are different because they don't share a common evolutionary origin. Yet the logic of the design is always the same. But that actually allows to study clock function in an organism like fungi and extrapolate what we learn to mammals and vice versa. What do we know about the clock in a fungus like Neurospora crassa, which is the main model that we use in our lab? We know that the core clock mechanism is composed of that diagram there, which is a negative feedback loop, where in blue you see what we call positive element, transcription factors that drive the expression of a gene called frequency. And frequency, we call it a negative element because it's going to be able to go back and inhibit the white color complex, shutting down its own expression. And that's what we call transcription, translation, negative feedback loop. That leads to oscillations in the negative element levels. But actually, you can take the DNA elements that control freak expression and hook it up to control luciferase, the gene that encodes for the enzymes that allow fireflies to have bioluminescence. And you can put that in Neurospora, and the movie should be playing. Oh, thank you. No, didn't play. You would see light coming up and down, and that's the quantification of the light signal in the graph on the left. But basically, you can have a bioluminescent fungus that is going to be showing you these molecular rhythms. Very importantly, the white color complex contains a domain called LOVE for light, oxygen, and voltage. That LOVE domain can sense blue light, suffers a conformational change upon blue light, that leads to love-love interactions, and now this complex can recognize a different type of element in the DNA and can boost up gene expression of frequency, for example, and that changes the amount of freak and in a way that changes the shape of the oscillation, and you can synchronize the clock to a new local time. And you can see there in the graph the arrow is where we provided the light pulse, and you can see that burst of luciferase activity and then the oscillation again. What are we doing with fungi? We're doing a lot of the understanding the molecular basis of circadian clocks. But to resonate to some of the talks throughout these days, we have been also interested in climate change. And we have been asking how having a clock might help an organism, like a fungus, to adapt better to climate change. So just a few quick words on this. This is a temperature record throughout months in a city in India, and you can see that in the summer month, it gets very, very hot. And I'm showing India because we all know that heat waves have been an issue there, but also because you can find Neurospora crassa, this fungus, you can find it in India. But even if you have a heat wave, during the night, it's going to cool off a couple of degrees, 10, 15 degrees. So even during heat waves, you will have oscillations of temperatures, hot days, cooler nights. And many times when people study thermotolerance, they somehow skipped this idea that when organisms are exposed to heat, they have the ability to cool off during the night, and that might help. So what we have been doing is doing experimental evolution of Neurospora. We can grow millions of individuals. It has mitotic cycles that go very fast, so we can have a lot of mitotic divisions in a short time span, and ask whether, if we expose Neurospora to very stressing conditions, like very hot days and cooler nights for many days, this is about 6 months of experiments, and measure growth rate, for example, you can see that about generation 1000, you have an increase in growth rate, which is telling us that the fungus is now able to grow better under this condition. It has better fitness because somehow it has coped with this constant stress, and there are some genetic changes that are underlying that. Now we're trying to understand what those genetic changes are. Why this is relevant? Well, colleagues like Arturo Casadevall at NIH have been proposing this hypothesis of climate change driving the adaptation of fungi to higher temps. Fungi in general are really bad human pathogens, but examples as Candida auris are really raising the flag that this might change. Candida auris could be found in the environment, was not really good with dealing with temperatures over 37 degrees. Yet, many of you have heard Candida auris breaks throughout the world. One of the characteristics of the clinical isolates is increased thermotolerance, and Casadevall and colleagues have been proposing that this is actually driven by the climate change pressure in the environment. So using Neurospora, we're trying to understand how actually these cyclic changes in temperature might be driving this. So for the remaining of the talk, I wanted to talk about light, not in the context of how to control organisms, but actually to understand basic processes. So for many years, Neurospora has been a great organism to study photobiology. We know that upon light, the white color complex, because of the love domain, has this conformational change and drive the expression of genes containing light responsive elements. So love domain is key. So as the song goes, all you need is love. Love is all you need when it comes to photo responses in Neurospora. So we can hook up a promoter like this to control luciferase, and we could measure how much luciferase do we get if we expose Neurospora to this much light or to this much light. But if you see the photograph on the left, you see that you can grow Neurospora on a Petri plate pretty much like a canvas, like a 2D surface. So one can start asking spatial questions when it comes to light responses. So in that video, if you see, I'm playing with a laser, and I did a drawing. For some reason, the movie was inverted, but if you were to look like that, you would see that the shape should be like that, okay? So the question is, was Neurospora able to see that very fast beam of light and mount a genetic response, a photo response, luciferase levels going up in the same manner? And you can see that surely it does. But the experiment looks quick and dirty. There's only one light intensity, and spatial resolution looked okay. So we wanted to know whether Neurospora, endogenous photoresponsive system, was able to detect more complex light pattern. So when you look at an image, if you use a projector and you project an image, you're actually projecting a map of light intensities. So that is actually a map of light intensities. So the question we had was whether Neurospora would be able to detect all the information when it comes to light intensities in a picture like that and reproduce it as a genetic program of luciferase levels. This is another way to phrase it. Can we turn a map of light intensity into an accurate map of light control gene expression? Or can we reproduce a picture with great resolution based on a dynamic gene expression system? So we started growing Neurospora on bigger dishes, like what you would find in the kitchen to make, some shepherd's pie or whatever. And we grow Neurospora in a very sort of like dense, thick manner. And then we use a projector to focus an image on top of there. Looks low tech, but that's how it works. We have the projector focus on the image there. And for the sake of playing with words, we call it live canvas because that's actually a living surface, a fungal mat where images can be perceived, genetically processed, and then emitted back as bioluminescence. And then you can capture it with a CCD camera. So we use an iconic image that people could recognize. And I want to stress this out. We were shining the image for only 1 second, okay? And then we wanted to know what Neurospora had seen. And that's the response that we see. Exactly. We have an accurate response that is recovering every information that was in the original picture. So we have a live reproduction of a picture based on a gene expression system. Some of you might be looking at this image and might be thinking, looks nice, but it's a little bit grainy. So the question is it grainy because of the biological limitation of our system or technical limitations in the way we do the experiment? So if you saw, we are capturing images every minute. And this is a very sensitive CC detector, but it's only a 1 megapixel CC detector. So then we moved to a Canon camera with a better CCD detector, but we had to use longer exposures, and that's what we get. So the resolution is much better because of the quality of the CCD detector, and moreover, you get the color of the natural luciferase. And what I like about this kind of picture is that you can see all the imperfections of the agar, and somehow those imperfections made the picture more perfect because of that itself. So this looks nice from an aesthetics perspective, but I also want to stress out that there is a lot of science within here. There is a lot of scientific questions that one can address related to transcriptional dynamics, photoadaptation, cell-cell communication, hyphal fusion, something that is at the core of fungal biology. And I like to show that picture of Winslow Briggs from Stanford, who was the first one to describe a love domain in Arabidopsis many years ago. So if you indulge me with the concept that Neurospora can see, okay, I want to play with that concept. Could Neurospora see the world if we just expose it to natural light, not forcing an image on top of it? So for that, we use a camera obscura type of concept, a pinhole camera, where instead of photographic film, we put the live canvas at the back, and then in front, we put this type of Canon camera that should capture whatever Neurospora responded of the light coming through the pinhole. Once again, low-tech technology looks like that in our hands. That's our camera obscura. And we chose a winter, gloomy afternoon to try to see whether Neurospora could see the world for the first time. This is from the top of our building at the Catholic University, and that's what Neurospora saw. That's ambient light going through the pinhole, and that's Neurospora responding to ambient light. We're not forcing an image. This is just what it's seeing through a small hole. So to finish, if you already indulge me with the concept that Neurospora can see, could Neurospora remember what it had seen? Could it have some sort of photographic memory, so to speak, eidetic memory? So as you already realize, we do circadian biology in the lab. So you can use a gene that is both light responsive and clock control, and you can put that DNA elements controlling luciferase expression. So if you were to monitor luciferase expression in the constant dark, you would see that the entire Pyrex should be oscillating in sync. So everywhere in the Pyrex should be oscillating with the same phase. But I also mentioned that light is a signal that helps synchronize the clock. So if you expose light in different places of the Pyrex, you're going to be shifting the phase of the clock. And the question is whether we can turn a map of light intensities into a map of phase changes. Once again, we use this image, exposing it for 1 second, and then we ask what happens with the circadian system for hours and hours afterwards. So at the beginning, we see the light response because this promoter is light regulated and clock control. And look what happens as we start monitoring over the hours. You see the image, it goes away, and you have the negative of the image. And then towards the next day, the image comes back to life and goes away to once again show the negative, and the next day, the positive. The agar is drying up, but the system doesn't care, it keeps on oscillating, because what we did was a perturbation of phase coherence within the entire culture, and this perturbation lasts a lot because of the quality of the rhythms of the Neurospora clock. That's another example. And it goes away, and now you'll see her coming back the next day. And there she is. These are just snapshots to show how it looks like throughout the days. So of course, we're doing a lot of circadian questions in this system, trying to understand something that is called phase response curves, phenomena like singularity, and so on and so forth. But because we are here, I also want to finish with this, how we have been turning this into also a way to do scientific outreach. So we recreated the Turin shroud with Neurospora. We used that iconic image of the Turin shroud, an artistic interpretation of it, and we used that to excite Neurospora for 1 second. And that's the end result. So many of you might be realizing that one of the things that stands out is the shape of the cross. And I wouldn't claim that this is a miracle. It's just that, it's just that there is more light information within that part of the picture of blue light, which is Neurospora actually seeing. So this, actually, we turned it into an institutional gift, and we presented it to the Pope. So this gift was actually based on science. And as I said back then, it is really thrilling to know that somewhere in the Vatican, there is a photograph of a photosensory biological response, in my case, of my favorite organism. So as our colleague said here, I didn't do any of this. It was the people in my lab who did it. Also in collaboration with amateur photographers that get excited with this crazy idea that started as an outreach project and then had a life on its own. So with that, I thank the Academy for this opportunity to present my fascination about fungi.